Last day will have to be a selfie day. Okay, so good morning. Welcome to day 4. So yesterday we were able to go through analyzing data and we had you obtain maps. The call for today is, what did we have you do? What did you have? What did you obtain? And part of the next set of lectures, we'll look into that a little bit more. Our first speaker of the set is Dr. Rich Hite, coming from the structural biology program at Memorial Sloan Kettering Cancer Center. He's worked with membrane proteins and ion transport, and something that has been really useful in his previous studies is that he's been able to analyze different conformational states from data and look at the validity of maps. And so something that we'll talk about hopefully in the roundtable is, now that you have a map, what's the next step? Map is not one and done. It's just the beginning of your cryo-EM journey. And the most important part is, Rich's favorite ice cream is black raspberry. Okay, Rich, you have the floor. Black raspberry.
Okay, well, thanks everyone. Thanks for coming. This is one of the more enjoyable lectures that I've been giving over the last few years. I've always enjoyed coming up here, first when I was a trainee and then more recently as an independent investigator, because when you think about cryo-EM in the US, this is really one of the centers, and the ability to come and discuss with your colleagues here and colleagues from other institutions just makes this an outstanding place to work. And so the way that it's developed into a national training center, it's really an ideal situation to come and get, for some of you, first training, for some of you more higher order training in cryo-EM. So please stop me at any point during this. I want this to be as interactive a lecture as possible. The goal is to make it a discussion rather than for me to lecture at you for an hour. That's not particularly rewarding for you. And I'd rather have a discussion than just talk at you. So any questions, everyone has a slightly different background, everyone has a slightly different perspective, so please stop me at any time. And so the goal of today is to go over what I think are some of the interpretations and limitations of single particle analysis. And as many of you know, this is a rapidly evolving field. So when I was a graduate student with Tom, the limitations of single particle analysis were very different from what they were, say, 3 or 4 years ago, and they're quite different from what they are today. So all of this are guidelines, not specific rules, because things are constantly changing, and what one can do with single particle, what one can interpret, is changing. And so the most important thing, and I think that hopefully I come back to it multiple times during the course of my lecture, is always go to your data. Always let your data guide what you're doing, because a number is essentially meaningless in a void, and only what you can see in the maps, only what you can see about the part of the protein that you're particularly interested in, can help you understand what your data is telling you. Okay, and so I think over the course of this week, you've had multiple lectures on how one takes a set of cryo-EM images, in this case of a bacterial or a clamp complex. You then do some sort of 2D averaging to demonstrate that you have well-ordered particles. These are well-behaved, they're monodisperse. You can align them. There seems to be at least some homogeneity amongst the sample. And then from that, you can generate some sort of density map. But really, as far as the biological understanding of cryo-EM, this is kind of the beginning. This is where you start generating your hypotheses to understand whatever it is that you're trying to do this cryo-EM project for. All of the stuff before it, all the biochemistry, all the molecular biology, all the structure determination is essential to getting to this point. But in order to generate important biological insights from your system, starting at the map is where you really start to move forward. And so for me, always the first question that I ask anyone in my lab and anyone when I'm in a meeting is, what is the question that you're trying to address with single particle analysis? Why are you trying to determine a structure? What are you hoping to learn? Are you trying to learn something about a mechanism? Are you trying to identify new components? What is it? And that will be essential for how you do all of your downstream analysis, because you're going to focus on trying to answer that particular question. You'll probably find other things along the way, but if two people may have the same set of data and may ask slightly different questions, and so they're going to interpret it and analyze it in slightly different ways. Second question is, what evidence do you have that supports the hypothesis that your structure faithfully represents the protein or complex that you wish to study? And this comes back to our first question, is that if you're looking for, say, an active state of a particular complex, and you don't know anything about how the complex works, it can be very difficult to ascertain what particular functional state your complex may be in. And so you may need to do your single particle analysis multiple times to get an ensemble of states that may give you some insights. Or you may know that you can add an inhibitor that locks it in a particular conformation, and that can give you some insights into what that state might be. Alternatively, you may have some orthogonal approach, some in vitro biochemistry or something along those lines that may help you to demonstrate that what you're looking at, what you've determined a density map of, corresponds to some important biological question, some mechanism that you're trying to understand. And the third question, and this for cryo-EM is something that most people don't necessarily recognize when they get started, is the domain that you're interested actually well resolved in your maps? In some cases, you may be looking at a disease mutant that was identified in some GWAS or some other human analysis, and you go through and you determine an amazing structure, 2 angstroms, 1.8 angstroms, and the residues that you're interested in are totally disordered. They're just not well ordered in the map due to either flexibility, conformational heterogeneity, or because they're an intrinsically disordered region. And while you may be able to get some insights into their function from looking at the rest of the protein, you're not going to be able to identify exactly what those residues are doing because they're not there. And this is important to keep in mind when you're doing your analysis, that just because you have a great map doesn't mean that it's going to answer the question that you're looking at. Another thing that can be important is if you do this and you have a particular domain or residues that you're interested in, and in your initial map they're not there, it doesn't mean that they're not there in any map. It may be that they're conformationally heterogeneous, and that's why they're important functionally. And so you may need to do classification or some of the other techniques that I think you've gone over this week to be able to identify different states of that domain, and that may help you to understand what that domain may be doing and why mutation of it may be detrimental for protein function. And for example, in this is a subcomplex of this larger complex here, and you can see where I've dotted around in the black dots, there's a whole domain that we were actually interested in understanding, but it was just not there. It was totally absent in the consensus reconstruction. Only when we did focused classification and focused refinement could we see where it was. And at that point, it was still lower resolution than the rest of the reconstruction because it's quite heterogeneous, and that was actually important for its function. So in some cases, this static disorder or this conformational heterogeneity may be really important for understanding mechanism. And it may not necessarily be a detrimental thing. If you can get even a lower resolution model of where it might be, that can help you dock in a higher resolution structure from maybe another group or something you've done previously and put that together to generate hypotheses for the mechanism. Another thing that can be important is orthogonal experiments. So can you do some sort of functional analysis? Can you use your structure to generate hypotheses that can then be tested experimentally, which one gives you more evidence that whatever you see in the structure represents the actual protein, and two, gives you more insights into the biological role of the protein. And so this is a structure of the pore of an ion channel that we worked on, and you can see there's a number of non-protein densities. And we did our best to try to assign these as water ions, but with cryo-EM density, we don't have anomalous scattering. We don't have the ability to describe exactly what corresponds to what density. We can do our best, we can make good hypotheses, and likely these are mostly correct, but it's not a foolproof approach. And so what we can do then is look for distinct features. And so in this case, we see that there's water molecules in this region, water molecules in this region, but where these two isoleucine residues are, we can see that it's mostly depleted of water. And so for an ion channel, being able to move ions through a hydrophobic environment is quite costly. And so that allowed us to generate a hypothesis that if we made it a little bit bigger, and so perhaps allowed a few more water molecules to pass through, it might change the functional properties of the channel. And so that's what this electrophysiological recording is showing, is that for wild type, it's a highly selective channel for potassium. When we mutate either of these residues to a valine, and so we're just removing one methyl group, now it's almost completely non-selective, and so it's equally selective for potassium and sodium, suggesting that this model that we've resolved, where we have waters above and waters below, probably represents what the structure looks like in a native environment. And so I've talked about this a few times, and I'm sure you've gone over this many times over the course of the week, is resolution. But what does resolution actually mean? And so when you think about, say, a 10 to 30 angstrom structure, which, say, you're doing tomography or negative stain for something that's quite heterogeneous, or it's a big molecule and you're just getting started and you're trying to see if you have the components that you think you have. So at 10 to 30 angstroms, you generally have, and this is the old era of blobology, where you have big domains, hopefully, that you can maybe fit crystal structures or other EM models into that can give you an idea of maybe the architecture of your complex. But at this point, you don't see secondary structure. You definitely don't see side chains. You don't see ligands. You don't see ions. And so this can give you, if you have many different conformations, in some cases, a starting point to work from. As you move to slightly higher resolution, say 5 to 10 angstroms, now you're starting to see secondary structure. Here you can see alpha helices. In some cases, you can see beta sheets. You're unlikely going to be able to separate individual beta strands, but at least you can see where a beta sheet might be, and that can give you an idea, particularly if you already know quite a bit about what the fold of the protein is. Then once you get past 4 angstroms, now you can start to move into the atomic modeling, where if you can build a de novo model into your density map, at 4 angstroms, you still need to be very conservative. It's very easy to make errors at 4 angstroms. As you approach 3 angstroms, it gets easier. At 2.5 angstroms, it's pretty unambiguous at that point. You can almost register your protein just by looking at the side chain densities. And so depending on where your model is, where your map is, you may be anywhere along the spectrum. And in some cases, as I pointed out before, one model or one map may have different regions that cover this entire spectrum. You may have some regions where you can see ligands, where you can see ions, and then other regions where it's more flexible. It may be more in this 5 to 10 or even the 10 to 30 angstrom regime. And so you need to be able to make your analyses and make your judgments that you're basing on those analyses in the light of these different regimes. And so does anyone know how we calculate resolution in EM? Has this been described this week? Anyone want to volunteer? Tom?
I guess not, Rich. You didn't do FSC curves and...
We didn't formally do FSC. We talked about it, but no lecturer went over it.
Okay, great. Well, then that's a critical thing, and I'm glad that I asked the question. So when you collect a data set, generally now it's millions or hundreds of thousands of particles. And each one of these should be an individual image that's independent of all of the other images. They're generally often from the same micrograph or from the same grid or from the same data collection, but each image should be an independent, unique view of your protein complex. If you then take your 500,000 or whatever it may be and split it into 2 halves independently, 250,000 in A and 250,000 in B, you then generate reconstructions entirely independently from A and B, so that they essentially haven't seen each other. There's no crosstalk. There's no information other than perhaps the masking parameters that are used. And that does introduce some bias, but it's quite weak, particularly as you get to higher resolution. What you can then do is calculate the cross-correlation between the particles in data set A and the particles in data set B. And the reason why we call it a Fourier shell correlation is when you do the cross-correlations in reciprocal space, the easiest way to do it is to take shells. And so you maybe take everything between 2.5 and 2.7 angstroms and compare them between the 2 maps. You then go between 2.7 and 3 angstroms, compare those between the 2 halves. And you can do this incrementally in higher and higher resolution shells. And as you are able to get higher and higher correlations, which are generally your lower resolution features, because these should be well aligned, as you get to higher and higher resolution, you start to see a fall off. And as you see this fall off, that corresponds to the resolution of the map. And so where you're able to get a high correlation between these 2 half maps is generally how we define the resolution. And the criterion that most people use is the 0.143 cutoff. And this was determined by Richard Henderson, now 15 years ago or so, to evaluate how one should calculate the resolution of a map. And in some cases, as I've brought up multiple times already, that may correspond to the mean. And you may have areas that are much more well-ordered and much worse ordered than that. And so while this is a useful criterion for thinking about your data, it doesn't necessarily work at the sidechain or domain level. And so there's additional tools that I'll come to later that can be important for thinking about resolution at a local level.
So what measure are you actually correlating is question 1. And question 2 is, I guess, the shell that you're expanding is around the electron density map that you're generating. And I guess where do you start that threshold?
So you don't think, generally when you generate a reconstruction, you have some volume that encompasses what you think is your protein density. Then you can put a soft mask around that. It shouldn't cover just the electron cloud. It shouldn't cover it shouldn't be anywhere near that close. It's usually 10 or 15 angstroms away from your nearest modeled atom, if not further. And so you have this very diffuse density shell around your volume. Then what you can do is calculate a Fourier transform of map A and a Fourier transform of map B. And because these are real objects, we get both phase and amplitude information. We can then do a cross correlation of the phase and amplitude information on a shell by shell basis. And so and that's one of the reasons why we do it all in reciprocal space is cross correlations of phases and amplitudes are quite facile for computational approaches in reciprocal space, and that gives you some value. If you have perfect correlation, say you have a synthetic data set where you've taken 2 copies of the exact same particles, your cross correlation should be 1. If you're taking protein complex A and protein complex B that are nothing alike, try to do a cross correlation. You may have some correlation at low resolution because you have some volume that just says you have density here but not density there. But as you get to higher resolution, so as we've talked about, even 10 to 30 angstroms, where you're thinking about domains. The domains for 2 independent complexes shouldn't align at all. So you have very low alignment there, and so your Fourier shell correlation will drop off very quickly. If you have 2 very similar complexes, say one with a ligand and one without a ligand, you may have a high Fourier shell correlation to a certain point, but whatever the conformational change is induced by that ligand, that will cause a fall off of the Fourier shell correlation at whatever resolution you're starting to see differences.
Take the soft mask at 20 angstroms out from your closest electron, and then you calculate the A and B, then you calculate the cross correlation of the Fourier transform.
Yep.
Then you shrink the mask down to 15 angstroms.
No, so in reciprocal space, now you have because you have a 3-dimensional volume, you have a 3-dimensional volume in reciprocal space. And so you don't need to mask that. All you're doing is taking a shell, taking all of the phases and amplitudes in that shell, comparing it with the phases and amplitudes of the shell from the other volume. Then you step out just a little bit further to the next shell, do the same thing. To the next cell, do the same thing. And generally we do this on a voxel by voxel basis. So if you collect a 256 by 256 pixel image, you have 256 on 3 sides for your voxel. And so that will tell you essentially how many discrete pixels you have. And so you would have about 128 bins in your Fourier shell correlation because you step out from the origin one at a time. The origin in reciprocal space is the center. So that would be 128, 128, 128. So you can go to 128 before you're at the edge of the volume. Tom?
You do this on electron density?
No, you do it on the phases and amplitudes, which are cool.
Yes, without the electron density, that's not what we really have.
Right.
The Coulomb potential.
Cool out, yes.
So electron density comes from X-ray crystallography, where the X-rays will measure the density of electrons. The electrons that we use are charged. They interact with the electrons also in a charged way. So what we actually, our density is called shielded Coulomb potential. Nobody wants to say that, so we just say density. But it's not electron density.
And they have slightly distinct properties, but until you get to extremely high resolution, it's not something that we generally worry about. We use the same tools for modeling and refining maps. Yes?
So how is local density figured out? Is it just that you shrink the area that the maps gets covering?
So that's essentially what you do, is you go through and do a cube by cube and calculate the Fourier shell correlation for each cube. Did you have a question, Tom? Or statement?
So you use electron scattering factors rather than X-ray scattering factors?
Yes.
So there's a slight difference.
Right. Right.
So now that I'm understanding it, 0.143 seems like a very low threshold because that means it's very low correlation between the maps A and B.
That works out to about CC 1/2 of about 0.5. So a cross correlation of about 0.5 because you have both phases and amplitudes, whereas for X-rays you only have amplitudes and not even amplitudes. You have the diffraction peaks. And so in the end...
So that is actually comes from, so I'm going to talk about the final result. It's a 60-degree phase error in X-ray crystallography. That correlates the Fourier shell correlation of 0 to 1. That's how this was introduced. It's a phase error of 60 degrees.
Yeah. Any other questions? Okay, and so while over the last now 8 or 9 years, the ability to obtain high-resolution structures has become possible. If you go into the EMDB and actually look at the statistics, and they update these weekly with every new set of maps that they release, you can still see that the majority of the reconstructions aren't particularly high resolution. So below 3 angstroms is a very small fraction, so still under 1,000 last year, compared to the lower resolutions where you see better than 16 is about 4,000. So it's still a small fraction or reaching this very high resolution. So while you often think when I start a project I'm going to get very high resolution, it's going to be very easy to do my analysis, this is why even today I still go through these descriptions, that most of the reconstructions are still in this 4 to 5 angstrom regime. And so you need to be able to evaluate your models and your maps appropriately at the resolution that they're determined. Another important feature is if you can look at the average. And so not just how many maps are at a particular resolution, but what is the mean resolution? And so it used to be well over 10 angstroms. And so up till 2014, you could see it was almost 20 angstroms. And this is where the new detectors came out, the new software really became robust, and this changed. And you could see since then it's been a pretty steady decrease in resolution, but the average is still not 3 angstroms. It's around 4.5. Sorry, my mouse is a little wonky today. And so most...
Is this just single particle?
This is just single particle. Yeah, you can filter it in the EMDB. So I every year before the lecture, you can see I did this the other night. I go through and look, because you can see it's getting better and better, but it's certainly not where we're getting atomic resolution structures on the average. The average structure is still worse than atomic resolution. And this is filtering out tomography, which are much worse, and electron diffraction, which is much higher resolution on average. And so, as I just mentioned, micro-ED, so electron diffraction of small three-dimensional protein crystals, is another way to use an electron microscope. It's not a single particle analysis approach, but it's a way to get high-resolution structural information. Generally, it's most used with small peptides. So this is a prion protein peptide sequence where they were able to obtain a sub-1 angstrom reconstruction. You can see, rather than these tubes or balls of density, here you actually see that there's individual ball spheres corresponding to each of the atoms of the molecule. And so this is an extremely high resolution map. We've started to come close to this with models, proteins with single particle analysis, but it's unlikely this is ever going to be routine. And now this is still better than average, this 2.6 angstrom reconstruction, again, of a model protein. But this is what one would hope to see. If you have a really good reconstruction, you're just a little bit better than 3 angstroms. You should be able to now see all of the side chains quite nicely. So here's a lysine, here's histidine. But one thing you can notice is that this glutamate here actually has very weak density. And this is common for glutamates and aspartates in EM density maps, is they tend to be lost more readily than any of the other side chain densities. So in some ways they can be useful for helping to register. But if you're specifically interested in the chemistry that these side chains are participating in, sometimes you have to really play with your density maps in order to see where the side chain actually is.
Is that just a result of the damage being smacked by the electrons? Because I know you run into that in X-ray crystallography a fair amount, where you start looking at things, it's a side chain, see your solid exposed or got radiation damage.
That's the most likely hypothesis. There's also slightly different interactions because electrons are charged, and so they interact with different side chains differently. But this has been documented at multiple resolution regimes. If you see something that's in a very well-ordered salt bridge, often the glutamates are still resolved. But if you compare them to, say, an arginine that's in a pair, as you threshold down, the glutamate will disappear more rapidly than the arginine, even though at that point it is more well-resolved. When they're floating out into solvent, then they're very difficult to see. Yeah. And so here's an example of a Fourier shell correlation. I thought I had one. And so this is now spatial frequency, so 1 over angstroms, and you can see that up until about 1 over 0.3 angstroms, so about 3.3 angstroms in nominal resolution, it's about 100% equal. So there's essentially no difference between the two half maps. And then it falls off, and then you can see by about 0.37 or 0.38, there's essentially no correlation. And they, in this map, or in this reconstruction, they actually calculated both the 0.5 and the 0.3. And they've generated their maps and filtered their maps using the 0.143 cutoff. Here is now a little bit on the lower resolution. So this is a 4 angstrom reconstruction, which was sufficient to build a de novo atomic model and start to generate insights into protein function. But you're not seeing side chains as efficiently. You're not going to have an easy job finding ions or ligands in the density map. But one of the things you can see is that even in this 4 angstrom reconstruction, there's substantial conformational change. There's substantial heterogeneity. So there's what they call the alpha-3 helix has 2 very different conformations. And by using 3D classification there, you can see some of the changes that it adopts. So here is more of a straight. Here the top part is mostly disordered. And here it's more of a bent conformation. And they were able to then make mutants based on this analysis that gave them insights into how this channel functioned.
Are there any mechanisms other than aligning all of those different density maps and then going by eye and identifying the different positions of that alpha helix?
Yeah, so we'll go into that later on. So this is a few years ago when you still had to do most things by eye, but now there's more sophisticated tools that help you to understand the heterogeneity in a more precise manner. Good question. And so resolution, what does it mean? What are some common features of samples that yield high resolution cryo-EM reconstructions? Anyone here have a benchmark sample that you know they use at your particular institute for doing, or what they use here? What? Yes. And do you know why apoferritin is commonly used compared to something like this, a GPCR complex?
It's very stable.
Exactly. And so if you think about most of the proteins that you're interested from a biological perspective, it doesn't have all that much in common with apoferritin. And so apoferritin is great for benchmarking a microscope. It's great for showing that you have a well-determined system and that your processing pipeline works. But as far as the kind of samples that most people are interested in understanding their function and their mechanism, coming back to my point earlier, is what is the question you're trying to address? Apoferritin doesn't have a whole lot in common. And so here's, for example, apoferritin. And so apoferritin is the highest resolution in the EMDB. I checked again, it's 1.15 angstrom. So not quite that sub 1 angstrom that we get from micro-ED, but not that far off. But if you go through all of the high resolution lists, and I did this, so 19 out of the top 20 reconstructions are all apoferritin. The other one was an adeno-associated virus. And so this is another highly symmetrical, well-ordered protein molecule. And so we just went over this. So what makes these good samples for cryo-EM? They're essentially rocks. You can leave them on the bench for weeks. They're highly symmetrical. They aren't damaged when you freeze them very readily. They're quite stable in the ice environment. And so they have a lot of properties that allow you to very quickly get many, many views of the particle. So when you do your reconstruction, you can generate a high resolution reconstruction because you have many, many copies that you can average together quite readily. And so why do many other samples not achieve this sort of high resolution? It's often because they don't share these properties. They're heterogeneous. They're often damaged by the air-water interface. They're not stable for a long term. So when you purify it, you have to very quickly freeze them. Oftentimes, the first, second, third, fourth, 10th attempt at getting a reconstruction, you could start, it'll improve, but often the biochemistry is limiting because you have a novel sample, a novel protein, and you don't know how to stabilize it in a way that can help you obtain high resolution. And so these are the sort of things that we just talked about. So low or no symmetry, difficult to produce, low concentration. You know, one of my favorites is you make a ton of protein, you put it on a grid, and you can see a bunch of protein stuck around the rims of the holes and nothing actually in the holes. And they all just get stuck to the carbon and you get nothing in the hole. And so go back and, okay, let's try a different additive, let's try a different detergent, let's try something to see if we can get more protein in the holes. But there's almost an infinite number of ways for samples to go wrong and very few ways for them to go perfectly right. And other samples, even when you get them nicely in the ice and they're well behaved, they're quite heterogeneous. Sometimes it's compositional heterogeneity where you have a mixture of different components. So particularly if you're doing something purified from an endogenous source, you may have 6 or 8 different subcomplexes in one intact complex, all of which are mixed in your particle mixture. Another is compositional heterogeneity. If you don't have, say, an inhibitor or something that can lock it in a distinct conformation, it may sample a whole ensemble of different states. And this will degrade your resolution because you're averaging all of these different states out. And then also is the idea, and some of these things are actually things that here at NCCAT, they're working actively on trying to build tools to help with. So weak interactions with either the grid or the air-water interface or preferred orientation, which you may have a great particle, it may all go on the ice, you may have really nice contrast, but you're looking at one view over and over and over again, and so you get great 2D averages, but you can't get a three-dimensional reconstruction. And so these are all issues that can be overcome, but they're really, really damaging as far as getting high resolution.
For example, concentration and heterogeneity or sample, is that something that you might just have a ton of data and a larger number of images, or is that just kind of not something you want to start with?
So it depends. Sometimes you can try, say, a graphene oxide grid, and if your protein absorbs and is not damaged by the graphene oxide, often you'll increase your local protein concentration substantially. You'll see more particles. There's other approaches where you, say, use an affinity tag. So you can functionalize the graphene oxide grid and have some interaction that can further concentrate your protein there. Others are, if you just collect a ton of data, sometimes that works, but often it's more beneficial to take the time to improve the biochemistry. Sometimes it just means instead of taking 1 liter of culture, you take 12 liters of culture and put all of your effort into making one perfect prep after you've spent months, years, whatever it is, getting your biochemistry optimized. Put that on a grid, and then you'll have one or a small set of grids that have a nice high density. And particularly for most of us, we have limited EM time. We can't just collect and collect and collect and collect. You can collect, and then someone else needs to use the instrument. So if you really put your time into optimizing the sample, often you'll end up with a better result in the end. Well, that's not a problem, I don't think.
To the point where it's too much.
So then reduce your protein concentration or try graphene oxide or some of these same things. These affinity capture, because there's some sort of fixed matrix, either the graphene oxide itself or the thin carbon or some sort of functionalized grid, those will force protein complexes apart, and you can use very low concentrations. In some cases, 2 or 3 orders of magnitude lower than what you normally have to use.
What if you try lower concentration, but then you get no protein?
In some cases, people use the mixtures. So you'll take something like apoferritin, mix it with your protein, and that can smooth out the ice in some cases, have a small amount of doped in per sample, and then a lot of your sample. In some cases, that also just buffers the protein-protein interaction so you don't have as much aggregation. Other times, changing the salt, changing the pH, changing other conditions about the buffer, changing your blotting time, changing the time that you apply the sample before you blot it. We have, there's almost an infinite matrix of ways that you can optimize grid preparation, and at some point it's quite challenging to figure out what's the next best approach. But generally, for some samples, we apply and we come back 3 minutes later and then we plunge. Other ones we apply and we have to plunge within a second, otherwise it aggregates. And these are all empirical, they're all stochastic, and so it's very difficult to come up with a general rule. But the more practice you have with your sample, the more you understand the biochemistry, generally the better you can get a sample onto a grid well behaved.
Is there some way to predict which target for EM will be more damaged by the air-water interface and which not? Because if I understand it right, one reason the apoferritin is so good is because it's basically not so much susceptible to this air-water interface damage. Is there something special about the protein that makes it basically not get damaged and other proteins do get damaged because they're more susceptible? Is there something
So
known about it?
I would refer you to a paper from Alex Noble, who works here at NCCAT, and he went through and did 24 different samples by tomography to look at the air-water interface, and everything goes to the air-water interface. It's just to what extent and how much it induces preferred orientation, how deleterious it is to protein fold. And in some cases, the air-water interface will tear your complex apart. In some cases, you get preferred orientation. In some cases, it's not such a big deal. But most proteins, because you have a very thin layer and proteins are rapidly bouncing, will eventually get stuck at the air-water interface. And so this is where some of the new tools like the Chameleon that's been developed here, where you're using a very, very tiny nanoliter drop of sample, and you're quickly wicking and freezing before it has time for all of this to happen, and it can be very helpful in some cases.
You mentioned affinity grids, which I used to be interested in. Is there now really a way that you can use this? Is somebody really succeed in making this a general?
I think it's still mostly theoretical, and I think it's still mostly hand-built, but it has the potential to do these sort of things. Whether or not, just like anything, it may work for you, it's likely not going to work, but if you're desperate, that's all of these sort of innovations are generally because people were desperate and whatever they had didn't work in their hands, and so they needed to try something else. And so I don't dissuade people from trying these things, but I usually try to start with the simpler things. Trying buffer, trying biochemistry is certainly easier than making some sort of affinity capture grid. But if there's no other way to do it, that might be a way for you to move forward.
You're saying that you can apply some protein integrated, you have to blot very quickly, otherwise they'll aggregate. Can you explain why that would be the case? Because I'm thinking you have a tube of protein that's been sitting there for at least a couple minutes. So why would they, and then once they get on the grid, start to aggregate quickly?
I think it's interaction with the carbon support in many cases, or the gold support. And so this is where sometimes switching from a regular carbon support to, say, an ultrafoil gold grid can make a difference because the support itself is different. You're also ending up with a much thinner volume, and so dehydration can occur, and that will change your buffer concentrations. Any other questions? Okay, so other ways that one can improve structure determination, as we mentioned, brute force, and 5 years ago, collecting millions of particles was not feasible. Now it's rather straightforward because with automated data collection and the high efficiency of the microscopes, you can often get 6, 7, 8,000 images in a day. And if you have a high density of particles, you can often get 1 million particles overnight or several million particles overnight. If you have a particularly small protein or very heterogeneous, this is where antibody fragments or nanobodies can be particularly advantageous because these are often conformationally specific. And so if you have something where you know it samples a whole ensemble of conformations, and GPCRs are a common example, where G protein GPCR complexes can be very heterogeneous and don't associate very well. But if you can have a nanobody that can lock either or both of the components in a particular conformation, you can stabilize that and obtain a higher resolution reconstruction. Small molecules are also great. If you know that a small molecule locks it in one particular state, then you're going to get rid of a lot of your conformational heterogeneity just because you bias the equilibrium with the addition of the small molecule. Sometimes proteins can do the same thing. You may have most of a complex and it's quite dynamic, but you're missing one component either because it hasn't been identified previously in the literature or because it's not that well associated. So maybe if you make a fusion protein to keep one component present at a higher local concentration, that can sometimes help stabilize your protein. Another is if you have a domain that you know is heterogeneous and it's causing aggregation, is sometimes just use molecular biology and cut it off. I know for cryo-EM, we generally try to keep intact complexes, the full construct, everything like that. But in some cases, that's the only way you can get it to work. And one of the great examples of cryo-EM over the last decade is TRPV1. And there still most of the work is done with a truncated complex because they know that there's a large domain that is intrinsically disordered, and by cutting it off, they can get much better reconstructions and allow them to gain insights into gating. They then go off and invalidate using a full-length TRPV1 construct, but by comparing the truncated and the full-length, sometimes you can get a higher resolution reconstruction that can give you more detailed insights, and then you may be able to validate that with a lower resolution reconstruction of a full-length complex.
So suppose you have a protein that half of the protein gives you a good, say, stable and rigid, but the other half is intrinsically disordered or has a very big roots based on the alcohol. Do you have any suggestion how to stabilize that in the food structures?
Sometimes these exact things. So antibodies, nanobodies, small molecules, protein cofactors, often these intrinsically disordered proteins are involved in protein-protein interactions. And so they only become ordered when they're involved in some sort of complex. And so it may not necessarily form a secondary structural motif, but it may wrap around the protein, and that could stabilize the fold enough that you can see parts of it. In some cases, if it's truly intrinsically disordered, cryo-EM is not really the approach. you're never going to be, cryo-EM is an averaging approach. And so you need the sample, at least the intrinsically disordered domains, to adopt a stable conformation that's reproducible. And if that never happens in solution, cryo-EM is not going to be able to force it into a distinct conformation. So you need to have some sort of exogenous stimuli that will change that equilibrium from something that's totally dancing around to at least limiting that to a smaller degree of freedom.
That being said, it's probably not feasible, but do you think if you took, like, say, millions, infinitely, like, large amounts of micrographs, you could create all the positions of density for those loops? I know the degrees of freedom would be the biggest limit.
I think if you're doing that, you might as well just run AlphaFold an infinite number of times and calculate the shell that might fit into, because both of them are essentially giving you an envelope in which that sample can move. And cryo-EM is not going to necessarily give you one that's more correct than AlphaFold, because in the end, all of these are just hypothesis generation approaches. You're not going to get an answer from an intrinsically disordered protein from either approach, but it'll tell you where it might be and what it might do that you can then generate hypotheses to evaluate with some sort of orthogonal approach. So I think both of them are viable. It depends on what, in some cases, AlphaFold doesn't give you anything useful at all, and so that's not helpful. So you may want a spectroscopic technique as well, where you can just see how far away can these two move. So if you have two loops and you can put a tag on either end, a fret pair or something, is it 10 angstroms away? Is it 50 angstroms away? Is it 500 angstroms away? How far apart are they going? And that might be able to give you an idea of what is cryo-EM ever going to work for you.
So if you have a very flexible loop and you take infinite data, do programs these days allow you to get all the different conformations? I wouldn't think so.
I haven't seen anything where that's worked yet. I think what you can do is, as you start to sample it out, you can see things that are more biased maybe to the left, more biased to the right, and get an envelope at very low resolution. But it's not going to tell you what the atomic constraints are of that protein, because there's just too little signal to noise there. And so this is a data set from a couple years ago we collected, just thinking about the number of particles as a function of resolution. And so from just about 500 particles, we could get a 4.5 angstrom map. As we went to 1,000, we got to 4.3, but you can see it very quickly becomes quite costly number-wise to get higher and higher. So 3.5 angstroms is 10,000, but 3 angstroms is 100,000. So we're very quickly getting into a logarithmic scale to get higher and higher resolution. And this is where eventually you either need a better camera, a better detector, or better biochemistry to push this slope. And so generally, you get to a position where you've essentially satisfied what you can do with the sample you have, and either if that answers the question, you're all set. If not, then you need to go back to the drawing board and think about how you can improve the data so that you can get to a higher resolution. And this comes to what I was saying a few minutes ago, that back in 2014, 400 images was a great day. That means your microscope was working well, it was aligned, you were collecting data the whole day. A few years later, it was maybe 2,000. Now, 7,000 is a reasonable number to be able to obtain with a Cryos. And so if you have a well-functioning facility, your microscope is well aligned, getting 7,000 images in a day. And so then it becomes density. If you get 2 or 3 particles per grid or per hole, you're still going to be stuck getting to large numbers. And so coming back to this idea of biochemistry and sample preparation, if you can get 500 particles, 1,000 particles per sample, and you get 7,000 a night, that's 3.5 million or 7 million there. And so you should be able to deal with a lot more heterogeneity there than you would have been a few years ago when you were getting 1/20th the number of images. And with the new next generation detectors, the signal to noise is so much higher that you may not need that 7,000 particle images. You may need 2,000 particles. And so in one day, rather than getting 1 sample, you might be able to get 3 or 4 samples. And so this is what I was talking about at the beginning, that the idea of, are there specific rules? No, because everything is evolving so rapidly still in this field that what worked a few years ago, that you would have had to collect 20 days to get 1 day now. And it may be with the next generation detectors or some new technology, software, hardware, whatever it may be, that may decrease again. And so we may move to more of a synchrotron approach, where instead of needing 1 person for a day or 2 to collect data, you may go and you get an hour, and that's enough to get a reconstruction, or a half hour. I mean, synchrotrons now, you can collect a whole puck of grids in 5 minutes, or crystals in 5 minutes. We may move to that eventually with EM, but right now we're not there. Right now, generally, if you need a large number of particles, you need about a day of data collection time. And so sample heterogeneity, we talked about this a little bit before, this idea of compositional heterogeneity, so where you have a mixture of different components or complexes with varying stoichiometries, as well as conformational heterogeneity, where you have one component that is adopting a range of different conformations with respect to the rest of the complex. And so often cases, these both exist within the same sample, particularly if you're working with a large protein complex. And these, up until the new detectors and the new versions of software, this was essentially an impossible problem to deal with. You really struggled with trying to sort these out. But now with Cryospark, Reliant, all of these tools can now quite effectively sort compositional and conformational heterogeneity. And so in some cases, you don't need the perfect biochemical sample, particularly if you're working with something as big as a ribosome, where your signal to noise is massive. With a small protein, it's still quite hard to sort these out effectively. And we've talked about this biochemistry multiple times: pH, salt, ligands, chaperones, protein-protein interactions, all different ways that you can limit the heterogeneity of your sample and thus improve the signal to noise of the particles you're imaging. And so for me, this was a paper now a few years ago, 2014, that really opened my eyes to what one could do with the software as far as handling heterogeneity. And so what they had done and what they were trying to accomplish was determine the structure of the mitochondrial ribosome. And this had been a long-standing question in the field. And one of the major difficulties was there was no established protocol for purifying mitochondrial ribosomes away from the cytosolic ribosomes. There's so many of both that it's quite difficult when you crack a cell to get just one and not both. And so instead of continuing to try to improve the biochemistry, they got to a point where they wanted to see if the computational tools were sufficient. And so they took a mixture of ribosomes from cells and just collected data. And they got about 100,000 particles. Again, this was now 8 years ago. Data was probably collected a few years before that. And they started to process it. And so they classified it out. And so they had a mixture of a number of different mitochondrial ribosome complexes. So some they're just the large subunit, some the large subunit and a small amount of density for the small subunit. Some for both the large and small subunit, and some for the large and small subunit of different conformation. But they also had a substantial number of cytoplasmic ribosomes. And by classifying these out, they could get purified just the mitochondrial ribosomes away from the cytosolic ribosomes. Then, because they saw that they had this substantial heterogeneity for the small subunit, they put a mask around the large subunit and said everything from the small subunit we're just going to ignore. Using this mask, they're able to then do a second round of classification and get a nice high-resolution reconstruction of the more rigid large subunit, get rid of some particles that didn't align as well, and then they were able to get a very nice reconstruction and build the first model of what the large subunit of the mitochondrial ribosome looked like. And this really changed the way a lot of people thought about single particle cryo-EM, because often cases you can't get a perfect sample. Crystallography has the advantage that you're driving things out of equilibrium into the crystal, and so if you have a mixture, a slight bit of heterogeneity in your sample, only the things that fit in the crystal lattice are going to be drawn into it, and so that can drive you out of equilibrium. Here, you're visualizing everything, and so if you have a mixture of components, you're just going to be stuck with that mixture. Now with these computational tools, you can actually start to sort them out. A few years later, Reliant became the next version of this, became more adept at sort of classifying. And so rather than just one or two components of classification, this is the Spliceosome, they're able to classify it on a whole range of different ways. And so they started out with now 500,000 particles, so a few years later you can collect more data. Through 2D and 3D classification, they ended up with about 140,000 particles that they thought actually looked like the Spliceosome. But the overall resolution is about 3.7, but they noticed that some areas of the map were much, much worse than that. And so they did local mass class refinement on different regions of it. And this improved the local interpretability and allowed them to build models of these domains. Then they used mass classification to try to see if within those local domains, could they find different states that might correspond to different functional positions. And so you could see they were able to get open and closed, different movements. They could see for this arm and the body, it was dancing around like this. And through iterative classification, iterative refinement, they could start to do rigid body fitting of better models into that density and get an idea of what it was doing. And so through this hierarchical and multiple tiered approach, they could take what was overall a pretty good map, but maybe not fully interpretable because local areas were too poor, and sort them out into different regions in which they could actually refine all of them to higher resolution and with improved interpretability. The next step of this is where they've taken it and actually automated it. And so this was coming back to William, is that your name? William's question of how much of this do you have to do on your own? And so now with what's known as multi-body refinement, you can just set volumes for different regions, and this will iteratively go through and do just a classification and say if your volume is an MRC. Just a classification on M, just a classification on R, just a classification refinement on C. And this allows you to move, say you have 3 balls and they can move up and down or back and forth. You can focus on M, focus on R, focus on C, get the best reconstruction of all of those. And then because you have the data from the particles in which they've come from, you can actually do Eigen analysis to see what sort of movements you're having with respect to one another. And so again, ribosome is a great sample because it's big, high signal to noise, and this is where they did a lot of their initial analysis. And this is taking a consensus map, and you can see a lot of the regions. This is local resolution, as we talked about before. The red is lower resolution, so between 6 and 5. And when they do the multi-body, you can see that the amount of red in the map goes down because they're able to better resolve those domains. But then they can also see what sort of movements are occurring within their data set. So these different eigenvectors correspond to the degrees of movement. And so the amplitude along eigenvector 1 is about 14% of the total movement in the data set. And it's a little bit easier to see in a movie. So here's eigenvector 1. And so you can see there's actually 2 different types of movement. The small subunit rocks, and then this head domain over here turns away.
It is great to understand movements right between domains, but how good is it when you really want to get high resolution of the individual domains? Do you do this, or do you do your own masking and refinement?
So often this works quite well because you do have, so this, and I didn't mention it, when you're trying to improve the local resolution, if you have a small domain that's moving and the rest of it is quite large relatively, you're still going to have, even if you put a mask around it, you're going to have contributions of the rest of it. So here what they do is what's known as signal subtraction. So they take only the area you're interested in and do a particle by particle subtraction to get rid of all of the rest of the density on the individual raw particle level. And this can improve your reconstructions because you reduce all of that background, which in this case, because you're only interested in, say, the M, everything that's the R and the C is actually noise, and you don't want to see it. It's signal because it's part of your protein, but in the case of a focused reconstruction just on M, it corresponds to noise. And so this can be quite helpful. In some cases, the difficulty is getting the precise map, and I think this is responding to Tom's question, that if you have the perfect mask, so you know exactly where your degrees of freedom are, you're going to do a better job. If you're just starting out with a big mask here, a big mask there, and a big mask here, it may not work as well because you may have multiple domains that are moving independently within the individual masks. And so in some cases it can be helpful. You can also do this exact same thing in a manual way where you do a focused classification refinement on different domains. And in some cases that works better. Depends on what your particular goal is. If you just want to see the overall movement of the complex, this works quite effectively. If you want to see a specific domain with very high resolution that you're interested in because that's your question, then you're going to want to do what Tom said and generate a mask just around that region and try a few different masks to really identify the best way to visualize those particles.
I was going to say, okay, so it seems like you're masking each individual domain separately, but then in the video, it's showing it all together. So how can you be confident that this conformation from this domain is present at the same time as this conformation?
So that's why they use this Eigen analysis, because if you have, say, 3 components, and so you have some n-dimensional space, and you can look at the movement of these 3 components, they look for the ones that are occurring simultaneously. So, for example, here they see this rocking here. That also corresponds to a rocking there in their Eigen analysis. If you look at a different eigenvector, it may be different. It may be, and remember, this is only about 20% or 18% of the variance. This isn't 100% of the variance. In some cases, you just have something like this, so that would be 100% of your variance. But with a ribosome, we know there's many, many different ways that it can move. And so one of them, the largest component, is this 2-degree movement. But the second eigenvector may be slightly different. And you can look at paper, or many other papers where they've done this, where often you have slightly contradictory eigenvectors because sometimes it'll be like this, sometimes it'll be more like this, and because proteins move in lots of different ways. They're not, so one domain may move in, say if you have 3 domains, domain A may move in the same way in every eigenvector, but B and C may move differently with respect to it. So A may only do something like this, but B may slide up and down, and so they don't have to all be the exact same. But generally the 3 components, so A, B, and C, each eigenvector, the relative movements of those should be distinct.
I think there was a misunderstanding. This movement with this is just one eigenvector. So that's just one movement. The movie that you showed at the ribosome, that's just one eigenvector.
Yeah.
So this is not all the movement, this is one of the movements, and you make a different movie for a different eigenvector, and so this is different.
Yeah. Yeah, of the composite model. It may be that eigenvectors 1, 2, and 3, one of those domains moves identically in all of them, but as a composite of the entire model, it's distinct.
So you would mask each domain separately and then put it together, or?
So this analysis then takes all of your broken pieces, stitches them back together, and generates a reconstruction. Then it also outputs these different eigenvectors so you can look at the movies of them moving. Yes?
So, in focus, you need a mask for your domain, right? So the mask should be tight enough to have only that domain and exclude the other ones. But it should also include the movement of that domain.
Yes, and this is where this comes back to Tom's point that it can be very difficult in a first pass, particularly when you don't know the degrees of freedom, where to draw your mask. You may think that sometimes a portion of domain A or subunit A may actually move with subunit B, and the rest of it may move independently. Particularly for things like ATPases that can undergo large conformational changes, they often have regions that tightly associate with neighboring subunits, and so it's almost, if you draw it on a subunit by subunit basis, your mask, you're going to miss it. You need part of one subunit, part of another subunit. That's your rigid body. And this is one of the limitations of this, is you have to have some idea of what sort of conformations you're looking at. And so another approach, one that it has a slightly different way, and this is done in Cryospark, is called 3D variability analysis. It's analogous, but rather than using masks to generate the different views, here you're just allowing the data to generate whatever conformational movements occur. So here I haven't drawn any masks, but if you compare different parts of the data set, you can see these substantial conformational changes. And so here you take a reconstruction, so all the particles have been aligned together, and you're just looking for the ways that it can move. This is a small channel, so the movements aren't very large, but if you take, they've done this also with ribosomes, and you can see tens of angstroms movements of domains, very similar to what you see in the multibody refinement, but rather than doing it on saying we need this mask here, this other mask here, and the third mask here, this is done in a mask-free environment, so you're allowing the data to tell you what it thinks is happening. And by doing them, they're often complementary. This can be really helpful for finding your degrees of freedom. And then if you want high-resolution reconstruction of the different domains, you can then go back to the multibody refinement, put a mask around, say, rigid movement A, rigid movement B, rigid movement C, and that will give you a better composite model.
So is the multibody refinement workflow, like, sort of under the hood, mask, give it a mask, and then it does 3D classification of those different conformations, then it merges that 3D classification back into a single model.
So multibody often isn't classification, it's refinement. And so this is the schematic of their architecture. So you have these 3 balls that correspond to 3 domains. If you want to get the best reconstruction of the M, what you do is you first subtract the density for R and C from your raw particle. You then put a mask around M, align it, reconstruct it, and generate a map of M to the best possible resolution. You then go back to the beginning and iterate it because you're able to get an improved alignment of M after that, and you can do this multiple times to try to improve it.
How do they pick out the big parts of M?
Because once you've eliminated R and C, if say M is doing this, if R and C are there, you're not going to move the whole complex because you're still going to be aligning on M and C. If you're just on R and C, if you're just aligning M and you get rid of R and C and say it moves 30 degrees, now what you can do is by reorienting the M, you can get M to align with all the other M's. So if you have some M's that are like this and some M's that are tilted over, if you align them in 3 dimensions, they're not the same. But if you rotate it, now they can all line up. Now you know the degree of movement that it has to go from the consensus from that. Yes, yes. And so that then generates these different multidimensional movements, because sometimes you have to move 30 degrees, sometimes 15 degrees, sometimes you tilt this way rather than this way. But they all come back to this consensus, and this is why you need to have the proper mask, because you want all of that movement to be encompassed by this one mask. So often you look at your, from the first pass, we use now 3D variability analysis because this can give us an idea of where the movements are. But if not, if you don't know anything about your protein, you can just look and say, okay, that looks like a domain, that looks like a domain, and that looks like a domain, and give it a try. If that doesn't work well, then you can try another set of masks. And you generally do this in Chimera. It's often the easiest way. You can use the segment map tool in Chimera to break out different regions of your volume, export them, and then generate a mask in either Reliant or Cryospark.
Can you just walk us through like the information that you pull out of this variability analysis?
Yeah, so the way that we use 3D variability analysis, particularly when it's, you see there's not 2 domains moving, the whole complex is moving is essentially one piece. So the way we would do this is we'd find the 2 extremes, say the 2 ends of the Eigen analysis for this vector of 3D variability. Use that then for a second class, so go back and do 3D classification. Start with this extreme, and see if we can pull out the particles that correspond to those 2 ends, and now generate reconstructions of those. So rather than going back and looking at the whole complex, in some cases, you may be, particularly if you're really interested in, say, what the head is doing, you may want to find out all the different conformations of the head and classify those out. If it's a complex like this, where it's just a simple channel, there's no large movements that have an entirely different eigenvector or a different movement degree of freedom. Here, you're gonna just try to take all of the particles and sort them out and see if you can get state A and state B from that, or maybe A, B, and C if you think there's 3 or more states. We go back and forth all the time, depending on what works. Sometimes Reliant works, sometimes Cryospark works. We usually start with Cryospark because it's faster, but Reliant often gives us more accurate classifications, so sometimes we'll do both.
I'm also familiar with this active. So you get this eigenvector. Do you actually, from the data, without doing the classification, can you figure out whether you have 2 discrete states or whether you have a continuous movement? Is there a way from the data?
Not easily, because what Cryospark does is it takes pools of particles along that continuum. And so you may take all the particles from, say, 1, 5, and 10. So your 2 extremes are 1 and 10. Your 5 and 6 would be your midpoint. But 5 and 6 may just be a mixture of 1 and 10. So it's difficult from that alone to see if you have discrete states. Whereas with 3D classification, often it'll do a better job where if everything is either state 1 or state 10 and you don't see anything in between, that would tell you that it's more of a discrete movement. But with small movements, the difference between a continuum and a set of discrete states can often just be signal to noise. How many particles do you have? How well resolved is that particular domain? Is your density just blurred due to local resolution, or is it because you have different conformations? So generally the way that we do it is once we have our final reconstructions, if we've sorted out A and B, state 1 and state 10, for example, we'll then take our model, build an atomic model of them, and then actually calculate, say, serine 32 to serine 32, how far is it moving? It's a little bit harder in Cryospark, or just in Chimera looking at the densities, to evaluate how far things are moving in actual angstrom distances. But if it moves just from a helix width to a helix width, you'd estimate that's maybe 6 or 7 angstroms. If it's less than that, it can be quite difficult. Yeah, so sometimes if your 3D variability, if your movements are small and the 3D variability analysis gives you nice density maps, that can be sufficient to calculate. You just take your model, fit it in, and then morph it into the two different states. And that can be enough to tell you how far things are moving. And so here's just an orthogonal view looking down the axis of the pore. And you can see that the pore lining helices are undergoing conformational change. And for us, this suggested that it had something to do with channel gating. And so we went back and generated reconstructions of both of these. And this turned out to be the open and closed state of this channel. The last thing that I want to talk about is some of the benefits of heterogeneity. So much of what we've talked about today is how you deal with heterogeneity when you want to get rid of it because you want to get a high resolution reconstruction. But there's actually information in that heterogeneity because we know biological molecules have to move. And this movement is often related to their function. And so this is a story that I did a few years ago as a postdoc with Rod McKinnon, where we were trying to understand the mechanism of activation of a sodium-gated potassium channel. And so we knew that from electrophysiology, as you titrated sodium, you increase the open probability of the channel, so the fraction of channels that adopt an open state. Structurally, though, we had a couple states, but it was difficult to know what exactly these states corresponded to. So one of the ways that we went back and tried to do this was to actually titrate the concentration of sodium in our sample, collect data, and calculate reconstructions of these. And so from the ensemble of particles, you can see most of them are this red state. And we have one that's this distinct, and this is the blue state. But you can see that there's a clear concentration dependence in this blue state. So at 20 millimolar sodium, it's very low, same at 40. It starts to increase at 80. And then by 160 millimolar, it's now the predominant state in our data set. And if we did a few more positions, you could actually do a dose response curve. So what is the fraction of particles belonging to this class as a function of concentration? And then we could compare that to electrophysiological analyses where we looked at the dose response of the channel open probability. And you can see they correspond quite nicely. And so this allowed us to use the heterogeneity to demonstrate that this open state that we've been able to determine structurally corresponded to the activated state of the channel functionally. And for things like ion channels, it can be very difficult to know precisely what conformation you're looking at. By using these sort of heterogeneous approaches where you're taking the heterogeneity, allowing it to inform what these states might be functionally, it can be quite powerful. I think that's all I have. Happy to take any questions. I know we have a few minutes left in the break. I went over by a little bit.
We have 20 minutes left in the break, and that's on purpose. So let's thank Rick.
Happy to take questions now or during the break as well. Yes?
So, you brought up ATPs as an example of a conformational change that you might be interested in. Something like this, especially in relation to an enabling supplement. But I guess a lot of them form homo-oligomers, right? And if you're aligning everything based on a conformation, is there any way to deconvolute, like conformational changes propagating around homo ring, or no?
It depends on is the movement symmetrical or is the movement asymmetrical. If you have two symmetrical states and that's all there is, say something like this and something like this, or it's rocking, then it can be very difficult. But if individual subunits are adopting different conformations, say the ring doesn't fire equally, you have some that have ATP, some that have ADP, and some that have a transition state along the way, you should get distinct conformations for those subunits. And so while your overall conformation will have a five-fold, six-fold, seven-fold, whatever your ATPase is, the individual subunits aren't going to have the same conformation. And so you can use that to get information. So something like the FO, F1 ATPase, where they have different positions around the ring, can have different ATP bound. That can be a way where you can, even though it's homomeric, the structure is different because of the different conformations and because of the different nucleotide bound.
And if there's accessory factors bound or a ligand bound, I guess you could use that as well to orient.
Depends on how big they are. If you know where the ligand is supposed to bind, then you can what is called symmetry expansion. So do we have a whiteboard or a marker? Great. So say you have 4 subunits, and you know that your ligand binds, or your protein-protein interaction partner binds out here. but you think it's substoichiometric. Maybe 25% have the ligand bound. Looking at, and maybe this is only a 5 kilodalton, it's just a small peptide. Very difficult to see on its own, but what you can do is, because you can align the entire protein, you can then say that this is A, B, C, D. You can then rotate it 90 degrees so that D becomes A. You can rotate it 180 degrees, C becomes A. Or you can rotate it 270 degrees such that B becomes A. Then you can, same sort of thing we've talked about, put a mask around it here, or maybe if you know exactly where it is, you can make the mask even tighter. Then you can do classification and find out which components or which of these protomers have your subunit bound. And then because you know, is it this one? Is it the zero, say your zero order? Is it the 1 turn, 2 turns, or 3 turns? Then now you know which of the particle orientations have this component there. Now you can go back and reconstruct your entire complex. With each particle, now you revert it back down to C1 because you know that it doesn't obey the 4-fold symmetry. So, but each component either gets a 0, a 1, a 2, or a 3 added to it. Yes, so you have 4-fold symmetry in your initial reconstruction, and then now what you're doing is you're looking at each of the components as individual molecules rather than as the entire 4-fold symmetric complex. Yes? And this is something that for ion channels we do quite routinely because they're often are homomeric complexes, but the subunits don't necessarily adopt the same